Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Rawal from Dr. APJ Abdul Kalam Government College, Silvassa, UT of Dadra and Nagar Haveli. Today we will discuss about inhibitors of electron transport chain under the title Carbohydrate Metabolism. So students, what we are going to learn in this module? Our primary target is to understand the concept of inhibitors and uncouplers. Our target is to understand inhibitors of respiratory complexes. We will also explore the inhibitors of ATP synthase. The mitochondrial electron transport chain is composed of series of electron carriers that operate together to transfer electron from donor like NADH and FADH2 to the acceptor such as oxygen. So this electron transport chain consists of carriers which carries the electron which is taken from the NADH, FADH2, and they travel along the carrier and finally it goes to the electron acceptor like oxygen. When final electron acceptor is oxygen, we termed it as an aerobic respiration. The electrons flow from carrier with more negative reduction potential to those with more positive potentials are eventually combined with oxygen and hydrogen to form water. The process by which energy from electron transport is used to generate adenosine triphosphate, that is ATP, is called oxidative phosphorylation. Many chemicals inhibits the aerobic synthesis of ATP and can even kill the cells. Obviously, if electron transport chain is inhibited, the cells will not get the sufficient energy and it will die. So, these inhibitors can even kill cells at sufficiently high concentration. These inhibitors generally fall into two categories. Some directly block the transport of electrons. These inhibitors does not allow the transport of electrons from one carrier molecule to the another carrier molecule of electron transport chain. The antibiotic pyridine competes with coenzyme Q. The antibiotic antimycin A blocks electron transport between cytochrome B and C, and both cyanide and azide stop the transfer of electrons between the cytochrome A and oxygen because they are structural analog of oxygen. Another group of inhibitors known as uncouplers stops ATP synthesis without inhibiting electron transport itself. Indeed, they may even enhance the rate of electron flow. Normally, electron transport is tightly coupled with oxidative phosphorylation. Do remember this? Oxidative phosphorylation involves what? The flow of electron through the electron transport chain and finally ATPase enzyme synthesis the ATP. So normally electron transport is tightly coupled with oxidative phosphorylation so that the rate of ATP synthesis controls the rate of electron transport. The more rapidly ATP, that is adenosine triphosphate, is synthesized during oxidative phosphorylation, the faster the electron transport chain operates to supply the required energy. Uncouplers disconnect oxidative phosphorylation. Do remember this, uncouplers uncouples the electron flow and synthesis of ATP. Uncouplers disconnect oxidative phosphorylation from electron transport. Therefore, the energy released by the chain is given off as a heat. So that energy is lost due to the uncouplers. That energy is not going to get converted into ATP, but it is lost, energy released, that is lost in the form of heat rather than the ATP. Many uncouplers like dinitrophenol and valinomycin may allow hydrogen ion, potassium ion and other ions to cross the membrane without activating a F1 ATPase complex. In this way, they destroy the pH and ion gradient. The force that is generated is destroyed. The valinomycin also may bind directly to this F1 ATPase complex and inhibits its activity. Concept map. So let's see how we are going to learn this particular module. In this module, our target is to understand first the inhibitors and uncouplers. The inhibitors mainly targets the complex 1, complex 2, complex 3, complex 4, while uncouplers targets the uncoupling between the electron transport and oxidative phosphorylation. Mitochondria generates adenosine triphosphate through oxidative phosphorylation in eukaryotic microorganism. But prokaryotes does not have a mitochondria. These steps carried out in the plasma membrane. Oxidative phosphorylation is the process whereby free energy that is released when electrons are transferred along the electron transport chain is coupled to the formation of adenosine triphosphate from adenosine diphosphate and inorganic phosphate. So in a nutshell, ATP is synthesized from ADP and inorganic phosphate. This oxidative phosphorylation is actually the main source of energy in aerobic cells. Oxidative phosphorylation generates the ATP, which is the currency of the cell. In uncoupled mitochondria, because electron transport may still occur, free energy may still be released. Over here, the electron transport is not coupled with ATP synthesis. So, the electrons are transferred down to the transport chain. However, the energy is not trapped as ATP. So, in such cases, we lost the energy in the form of heat. Mitochondria play an important role in satisfying the requirement of cellular energy through oxidative phosphorylation. Apart from this, it plays many other important roles. It acts as a buffer. It buffers the cytoplasmic calcium. It creates and liberates the reactive oxygens. It releases the metabolites which control crucial pathways like succinate and alpha-ketoglutarate. It helps in controlling the apoptosis. It assists the cell to adapt change in substrate availability by signaling pathway. They modify their organization and dynamics for control. So, the role of mitochondria indicates that plays a vital task in maintaining the balance within the cell. It is very important to maintain the harmony within the cells. It requires a strong regulation. It requires the control, the regulation of mitochondrial activity through expression, transcription, and translation. The mitochondrial activity and the enzymes are controlled at each level. Recent studies indicate that Regulation of mitochondrial activity is also achieved by dynamic assembly, organization of the respiratory protein complexes in the mitochondrial membranes. In Module 16, we learn about the respiratory assemblies. Over here, we discuss that task. We discuss about the free-floating model versus the super-complex assembly. We also conclude that as and when we may have a more conformational proofs, we may come to cure certain diseases related to the dysfunction of the mitochondria. 5 protein complexes of the mitochondrial respiratory chain. Complex 1 includes the NADH ubiquinone oxidoreductase. Complex 2 includes the succinate ubiquinone reductase. Complex 3 ubiquinone cytochrome C oxidoreductase. Complex 4 that cytochrome C oxidase, and complex 5 which plays a crucial role in the ATP synthesis, that is ATP synthase. Electrons are released from NADH2 that enters into the complex 1. Then electron is transferred through a series of electron and hydrogen carrier, that is complex 2 and 5. This transfer of electrons and protons generates the gradient. This gradient drives the synthesis of ATP through ATP synthetase, that is complex 5. So ultimately we learn during the glycolysis, the TCA cycle and the fate of pyruvate that these reactions releases the NADH, FADH2. This NADH and FADH2 releases the electrons to the electron transport chain. The movement of electron in the electron transport chain generates the gradient of hydrogen ion. This gradient actually drives the synthesis of ATP through the complex 5 ATP synthesis. The energy derived from the movement of these protons is used to synthesize ATP from ADP and inorganic phosphate. Formation of this ATP in such a mechanism is known as oxidative phosphorylation. What are inhibitors? So students, let's finally discuss the core aspect of this module. Inhibitors are the inhibiting agents. They actually bind with the electron carrier. They are specific in their action. The inhibitors binds to different component of the electron transport chain and they block the carrier. So from that point onwards, the transfer of electrons will stop. After binding with the carrier, it does not allow to change in a reversible form from an oxidized state to a reduced state. It leads to, and this results in the accumulation of reduced form prior to the inhibitor point and oxidized form of the component of the electron transport chain down the line of inhibition point. The synthesis of ATP stops due to cease of energy release. The energy will stop releasing and ultimately the ATP synthesis will stop. The most important non-inhibitors of the electron transport chain are amytal, rotenone, antimycin A, carbon monoxide, sodium azide, and cyanide. Site-specific inhibitors of electron transport chain have been identified. Actually, these inhibitors are very specific in their action. These compounds prevent the transfer of electrons by binding to a various complex of the chain, which blocks the redox reactions. Now, let's try to understand these inhibitors one by one. Let us first start with the rotenone. Rotenone is a plant product. It is extracted from the roots of the plant, Deris elliptica. It is an insecticide. It is a strong inhibitor of complex 1 of the electron transport chain. It inhibits the electron transport of electron through the NADH coenzyme Q reductase complex. It is reported that certain tribal communities use it as a fish poison By consuming this poison, the fish are paralyzed. This slide explains how rotenone affects on the complex. Next, we will start with the amytal. Amytal is a barbiturate. It inhibits the transport of electron through NADH coenzyme Q reductase complex. Demerol, that is another inhibitor. It also inhibits the complex 1. Quercetin A. It is an antibiotic of microbial origin. Its mode of action is similar to the rotenone. Next, that is antimycin. It is also an antibiotic from Streptomyces species. It blocks the electron flow at complex 2 of electron transport chain. It eventually inhibits the flow of electron from cytochrome B to cytochrome C1. Cyanide. It is a respiratory inhibitor which blocks the complex 4 of the electron transport chain. It blocks at cytochrome oxidase, which prevents both coupled and uncoupled respiration. Cyanide binds with the ion within the protein complex and prevent the regular activity of the complex system. It blocks the transport of electron to oxygen, which stops the further passage of electron through the electron transport chain. So actually cyanides prevents the transfer of electron at terminal point. They are analog of the final electron acceptor. As a result, the person is deprived of energy to carry out the many numerous processes that sustain the life, and the person dies. So in the case of cyanide, if a person consumes the cyanide, it will die because they don't have energy to carry out the routine processes. Similarly, carbon monoxide. It is also a respiratory inhibitor which blocks the complex 4 of the electron transport chain. It binds with the cytochrome oxidase, that is of complex 4, which blocks the transfer of electrons to oxygen. Carbon monoxide binds with the reduced form of iron in the heme group in cytochrome oxidase. That is again the part of complex 4. Azide. It is a respiratory inhibitor which blocks the complex 4 of the electron transport chain. It blocks the electron flow between the cytochrome oxidase complex and oxygen. It reacts with the ferric form of a complex 4 of electron transport chain. Hydrogen sulfide. It is a respiratory inhibitor which blocks the complex 4 of the electron transport chain. It is toxic. Now, let's try to explore the uncouplers. We already learned about the inhibitors. We discussed some of the inhibitors and their mechanism of actions. But the mechanism of uncouplers are quite different from the inhibitors. First of all, let's try to explore the concept of uncouplers. In intact mitochondria and in special preparation of submitochondrial particles, the transport of electrons and the phosphorylation of ADP are tightly coupled reaction. Over here, my friends, it means that when electron is getting transferred through the electron carrier, the transport is coupled with the generation of ATP. In damaged mitochondria, electron transport may occur through the electron carrier, but it is not coupled with the oxidative phosphorylation. When the transport of electron occurs, but the ATP is not generated through the oxidative phosphorylation, when this happens, the mitochondria are said to be uncoupled. Chemical agents which uncouple the electron transport with ATP synthesis is known as uncouplers. of electron transport chain. Such agents will stop the synthesis of ATP, but transport of electron will continue. It results into the loss of energy in the form of heat, since the energy will be generated, but that will be lost eventually in the form of heat. For example, dinitrophenol. These uncoupling agents are generally lipid soluble. These agents binds with the hydrogen ion and transport them across the membrane. During electron transports, hydrogen ions are pumped across the mitochondrial membrane, but this uncoupling agents carries it back to the mitochondria. So over here, the gradient will not generate since the hydrogen ions are carried back to the mitochondria. It will stop the generation of proton gradient. It will stop the synthesis of ATP by oxidative phosphorylation. Rather, the energy derived from the electron transport is released as a heat. So we learned that the uncouples the transport of electron and oxidative phosphorylation. The gradient is not generated. The energy derived from electron transport is released as a heat during uncoupling. Such kind of heat generation is known as non-severing thermogenesis. It is very important in many biological situations. In fact, in certain cases, it is necessity for certain organisms. For example, uncoupling occurs naturally in the brown adipose tissues. The inner mitochondrial membrane of such tissues contains a protein called thermogenin. This thermogenin protein is recognized as uncoupling protein. This thermogenin protein is act as an uncoupler. Due to their mode of action, there will be uncoupling between the electron transport and oxidative phosphorylation. Thermogenin is an endogenous protein found in the brown adipose tissues. Thermogenin act as an uncoupler which uncouples the ATP synthesis from electron transport chain by creating a passive proton pump within the inner mitochondrial membrane. Such tissues found in newborn animals and hibernating animals. So, presence of such tissues will help such newborn animals to generate the heat by themselves. These slides give us the example of certain uncouplers with the diagram. Over here, the diagram of that nitrophenol FCCP has been given. We already gone through the certain examples of the uncouplers in the previous slide. Chemical uncouplers are 2,4-dinitrophenol, dicumarol, CCCP, that is carbonyl cyanide metachlorophenyl hydrazone, and FCCP, that is paratrifluoromethoxycarbonyl cyanide phenyl hydrazone. CCCP, that is a carbonyl cyanide metachlorophenyl hydrazone, is a lipid-soluble weak acid. This CCCP is a very potent mitochondrial uncoupling agents. So basically these uncouplers are divided into two groups, that is chemical uncouplers and physiological uncouplers. Whatever the uncouplers we discuss so far, they are the chemical uncouplers. Physical uncouplers, we have the example of thermogenin protein, which are present in the brown adipose tissues, and they are very helpful for the newborn animals. Physiological uncouplers includes the long-chain fatty acids, thyroxines, thermogenins, that is a protein. These uncouplers have a hydrophobic nature which makes them soluble in the lipid bilayer of the membrane. This is very important. If they do not have hydrophobic nature, then they may not get into the target site and they may not exhibit their effect. So it is very important for them to become hydrophobic, so and so they can cross the membrane barrier. These uncouplers have a dissociable proton which allows them to bring protons from the intermembrane space to the matrix. It will disintegrate a pH gradient. This energy lost as a heat. So basically these inhibitors we can group into two. These inhibitors of the ATP synthase complex, that is the phosphorylation inhibitors, or the inhibitors of the ATP-ADP translocase, that is the transport inhibitors. Now let's try to understand these inhibitors of the ATP synthase complex, that is specifically which inhibits the phosphorylation. These inhibitors prevent the synthesis of ATP by binding to the ATP synthase complex. It prevents the inflow of protons, example oligomycin dicyclohexyl carbonamide. Oligomycin is a polypeptide in nature which acts as an antibiotic which is obtained from the Streptomyces species. It stops the transfer of high energy phosphate group to adenine diphosphate. Oligomycin binds toward 23 kilodalton polypeptide in the F base plate and blocks ATP synthesis by the ATPase F0 F1 ATPase, prevents respiration in mitochondria and all ATP driven functions in submitochondrial particles. Another phosphorylation inhibitor is a dicyclohexyl carbonamide, that is a DCCD. It makes covalent bonds with amino acid glutamate of the C subunit of the F0 base plate. When it binds covalently, it stops the proton channel which prevents the formation of ATP synthesis. Inhibitors of ATP-ADP translocase, that is, we said a transport inhibitor. Atractyloside, that is a plant glycoside, or bromgrigrate, an antibiotic from fungi, is able to inhibit the ATP-ADP translocase with very small concentration. Atractyloside is a plant glycoside. Absence of ATP also leads to the inhibition in the process of ATP formation. It is due to a tight coupling between the oxidation and the phosphorylation. It means oxidation cannot proceed via the respiratory chain without the simultaneous phosphorylation of ATP. So students, we learn a lot of things about the inhibitors of electron transport chain. Let's try to conclude what we have learned in this module. Electron transport chain inhibitors acts by binding somewhere on the electron transport chain. Literally, it prevents electrons from being passed from one carrier to the next. If the electron does not pass, obviously the gradient will not generate. If the gradient is not generated, it will not convert it into the synthesis of ATP. They all act specifically, that is, each inhibitor binds at a particular carrier or complex in the electron transport chain. Irreversible inhibition results in a complete stoppage of respiration via the blocked pathway. Competitive inhibition allows some oxygen consumption since a trickle of electron can still pass through the blocked site. Although it allows some oxygen consumption, competitive inhibition prevents maintenance of a chemiosmotic gradient. Thus, the addition of ADP can have no effect on the respiration. Whatever the mechanism of this inhibition, an electron transport inhibitor can block respiration specifically along the NADH pathway, along the succinate pathway, or along the pathway that is common to both routes of electron entry. Careful addition of inhibitors to mitochondria on specific substrate can reveal the sites of inhibition. Some combination of inhibitors enables demonstration of alternative entry point of the electron transport chain. So, by studying the mechanism of these inhibitors, we may come to know many different aspects of the electron transport chain. Thank you very much.